So I did my research with the Phycology Research Group in Kent University. So my thesis topic was phenotypic plasticity of the brown seaweed Dictyota dichotoma in response to climate change. So climate change is a global phenomena which is induced by the anthropogenic activities. So under this scenario, we can see several environmental changes. One is ocean warming. So sea surface temperature is predicted to rise by around 3 Celsius at the end of the 21st century. And it is an obvious fact that the marine communities get negative impacts due to the ocean warming. So they need to have mechanisms to cope with the changing environment. So in the marine environment, we can see several mechanisms. One is changes of natural distribution and also the possibilities to evolutionary adaptation. And also we can see phenotypic plasticity. So what is phenotypic plasticity? This means an ability of a single genotype to produce more than one alternative form of phenotypes in response to environmental conditions. This contains two major compartments. One is within generation plasticity. This means plasticity occurs in the individual's lifespan as reversible plasticity or developmental plasticity. And the other one is transgenerational plasticity, which means the parental generations transfer the plasticity to next generation according to the environmental changes. So for this research, we use Dictyota dichotoma. As the other marine communities, marine macroalgal community also get negative impacts due to the ocean warming. On the other hand, this organism is an ideal model organism because these organisms can be grown and manipulated easily at the laboratory conditions. And their life cycle is very special fact because they have sporophytes as well as dioecious gametophytes. On the other hand, this organism is a prominent seaweed because of their widened distribution in the European coast, also in the subtropic areas. And finally, there are many new research applications on this organism, and also the phenotypic plasticity, especially the transgenerational plasticity concept, is a new research concept on this organism as well as the marine macroalgal community. So our main goal of this research is to evaluate the role of phenotypic plasticity of brown seaweed Dictyota dichotoma against future ocean warming. So we did three different experiments in this research. First one is environmental predictability experiment. Second one is within generation plasticity experiment. And the third one is transgeneration plasticity experiment. So the first experiment, environmental predictability experiments. So here we want to find whether the environment of Dictyota dichotoma is variable and predictable to perform the adaptive parental effect. And the second one, within generation plasticity. So here our main goal is to find whether the ocean warming will negatively affect the growth rate of Dictyota dichotoma. And also we try to find the prolonged exposure to elevated temperature can partly reduce the stress response. Also, applying a thermal shock by 3 Celsius can increase the stress response compared to a thermal ramping. The third experiment, transgenerational plasticity experiment, our main goal is to find whether the parental effects can be transferred, especially the maternal effects on egg size, and then whether these mechanisms are adaptive. So how we did this research? The first experiment, environmental predictability experiment. So here we obtain time series data for sea surface temperature of goose through the Netherlands Water Information website. And then we clean the data, especially we choose the summer months for our analysis. And then we perform autocorrelation for consecutive days to develop the correlogram. And for the next two experiments, we use seaweed culture. So to prepare the seaweed culture, first we sample sporophytes from goose, and then through the spores of these sporophytes, we obtain gametophytes. Then we fragmented these gametophytes in order to make clones. These clones were introduced into two different temperatures. One is 22 Celsius, which is the optimal growth temperature for Dictyota dichotoma, and the other one is 25 Celsius, which is elevated summer temperature due to the ocean warming. And we use 30 milliliter glasses to culture the seaweeds in modified pro-vessel medium, and also we provided necessary light condition in every incubator. Then I move to the other experiments, the second one within generation plasticity experiment. Here we had 4 different thermal conditions, and we used the strains, actually the clones of same set of strains, which were grown in 22 Celsius and 25 Celsius. So we had the same temperatures, 22 Celsius and 25 Celsius, and also we introduced the strains which were in 22 Celsius into 25 Celsius with the thermal ramping and without thermal ramping. And we grew these strains in the particular incubator for 3 weeks, and then we analyzed the surface area of the strains using ImageJ software. Thereafter, we calculate the relative growth rate of the strain for week 1 and week 2. Here we use the specific growth rate equation. Finally, we perform the statistical analysis using repeated measures two-way ANOVA. Okay, so the transgenerational plasticity experiment, we use the gametophytes in 22 Celsius and 25 Celsius. Here, we introduce the red filters into their incubators in order to make fertile fronds. We use these fertile fronds to perform the crosses between the condition and within the condition. We wish to make fertilized eggs using these crosses, and also we had non-fertilized eggs to measure the egg size. So what is our results? The first experiment, environmental predictability experiment, gave this correlogram. So here you can see significant positive correlation between SSTs for 11 days. According to the reproductive biology of Dictyota dichotoma, they initiate and release gametes at regular biweekly intervals. So combining these two facts, we can say the effects of SSTs on offspring depend on the temperature of their parents' environment. Because the environment of Dictyota dichotoma is predictable and variable to transfer the changes of SST to the future generation up to 11 days. The second experiment, within generation plasticity experiment, we obtain mean specific growth rate of the strains for 4 thermal conditions. According to the statistical analysis, we obtained significant difference between the growth rate of the strains which were in 22 Celsius and 25 Celsius. This indicates a stress condition at the elevated summer temperature for Dictyota dichotoma. So the great reduction of the growth rate at 25 Celsius can be happen because of the cellular and subcellular damages, and also the reduction of the pigment production due to the rising temperature. This can lead the reduction of photosynthetic productivity. But we could not see any growth increment in 22 Celsius. So we think this may happen because they could not maintain the optimal nutrient intake because of their larger surface area. Surprisingly, we could not see any difference between thermal ramping and thermal shock experiment. So this suggests that the sudden increment in temperature by 3 Celsius may not be a huge shock for them. But we could see that the thermal shock and thermal ramping conditions, strains grew better, especially they got higher growth rate in the first week. So this can be happen because they have natural reservation of fatty acid, lipids or the nutrient which help to cope with the stress environment. Also, there may be a formation of heat shock protein when they are exposed to the higher temperatures. But we could not see any growth increment in the second week at these temperature thermal conditions. So it suggests that they could not maintain the growth rate in the second week. And I would like to show this graph also because it shows the growth rate of each strain in 2 different temperatures, 22 Celsius and 25 Celsius. And you can see some of the strains perform better at the elevated temperature. So this can be an indication of genotype and environmental interaction effects, which can be a possible selective outcome to be performed in the future elevated temperature through the phenotypic plasticity of the population Dictyota dichotoma. But to have a concrete conclusion on these results, we need replicates in each condition. And the third experiment, transgenerational plasticity experiment. So here we could not get any fertilized eggs. So we think this happened because the non-simultaneous gamete liberation from the gametophyte. So we need to continue this whole research. But I would like to show some interesting figures while I get during the experiment. So this is the reproductive structures of the Dictyota dichotoma. So first figure shows the oogonia on female gametophytes and then the antheridia of male gametophyte. And we could see this kind of egg liberation from the female gametophytes. And this is the picture of egg of Dictyota dichotoma. So what is our conclusion on the experiment? So the first experiment, environmental predictability experiment, suggests that the adaptive parental effects are performed by the population Dictyota dichotoma in response to ocean warming. And the second experiment, within generational plasticity experiment, strongly suggests that the ocean warming will negatively affect the growth rate of Dictyota dichotoma. As a general conclusion of our research, I would like to say the phenotypic plasticity may be beneficial for Dictyota dichotoma in terms of survival in the rising sea surface temperature at the future climate change. Finally, I would like to give some recommendation, especially for the transgeneration plasticity experiment. The first one is to have more tissues because it will enhance the probability of the fertilization by having more reproductive structures. And the second one is to provide night light because in the natural environment, Dictyota dichotoma release gametes simultaneously according to the lunar cycle. So these are my references, and thank you for your attention.